The sickle cell trait was supposed to be an adaptation to the human race of malaria. Those who inherited the trait became immune to the parasite, but when two people with the trait had children, the child would develop sickle cell disease. Sickle cell anemia affects those from African, Hispanic, Middle Eastern, Asian, Indian, and Mediterranean descent due to their susceptibility to malaria. Many are still unsure of why the parasite does not infect them, but hypotheses include the shape of the red blood cell and the increased phagocytosis. There are many clinical trials available today for those with sickle cell anemia, but one of the down parts of that is that it's hard to predict the effect it will have on the patient. Another major challenge with treatment is that many are diagnosed at a young age with sickle cell anemia, and as they grow into adulthood, there are less treatment options for them due to the lack of physicians who are untrained to treat sickle cell disease. The whole point of this project was to create a model of sickle cell anemia to test out different treatment options. To create this model, you would have to look into a field known as computational biology, which uses math and chemistry to show how our body works. This would allow people to test different treatment options without the use of human subjects. One of the possible things that could be tested with this model, and it's one of the things I found through my research, was called microfluidics. Essentially, microfluidics shows a relationship between pressure, viscosity, and oxygen saturation within the blood. So essentially, the higher viscosity and pressure within the blood, the less oxygen saturation there is. There are devices out there, though, that read the higher pressure, which could predict the place of an occlusion depending on where the pressure is. Another thing that I found in my research was something called reciprocytes. Another word for that is an artificial red blood cell. They are generally used in emergencies to raise oxygen saturation, and they are powered by the breakdown of glucose molecules and used for energy. They have three main binding sites within the reciprocyte: one that takes oxygen to the tissue, one that takes oxygen to the lung, and then one that takes carbon dioxide to the lungs to then be released through the body. Through this project, I was able to find that one day there are microfluidic devices that can predict where an occlusion will happen based on the pressure changes within the blood. Also, these things called SS2 cells, which are the cells that deform to fit through the different shapes and sizes of vessels, are what start the occlusion. People with sickle cell anemia form these sticky proteins on the surface of the red blood cells, and the proteins end up sticking to the vessel walls. This was discovered by a model that was created by Brown University. Reciprocytes may also be used as a long-term cure for long-term care for sickle cell disease, and maybe one day a cure. Overall, the cellular modeling can be used to test all of these without actually harming the human subjects. Overall, and bringing it all together, microfluidics may be able to predict where the occlusion will happen, and then the reciprocytes may be able to prevent the sickle cell disease. The tests using the models would be able to test all of these options, again, without using the human subjects, and could prevent complications when there's an emergency. Overall, the model was very difficult to create in the time given than I had anticipated. And overall, this project was more of a learning curve for me that showed me important skills like communication and time management. Thank you.